Assalamu alaikum wa rahmatullahi wa barakatuh. Hello everybody, hope you are all well. I am Dr. Dai Karim Metwalli, lecturer of histology. In this video, we are going to discuss salivary glands. What are our ILOs? By the end of this lecture, you should be able to enumerate the different types of salivary glands, describe the structure of the major salivary glands, stroma and parenchyma, correlate between structure and function of the major salivary glands, describe the structure of different types of the major salivary glands. Salivary glands are divided into main or major salivary glands and accessory or minor salivary glands. Minor salivary glands, they are groups of salivary glands having no capsule, present in the lamina propria of the mucous membrane of the oral cavity. They include lingual, labial, buccal, palatine glands. While main or major salivary glands, they are three-paired glands. They are compound tubular alveolar glands. They include parotid, submandibular, and sublingual salivary glands. The major or main salivary glands include parotid, submandibular, sublingual. The parotid gland located below and anterior to the ear on either side of the face, while the mandibular gland located between the mandible and muscles of the floor of the mouth. The sublingual glands located in the floor of the mouth on either side of frenulum of the tongue. Salivary glands secrete saliva. It's the major function of salivary glands. 70% of the saliva are secreted by submandibular gland, while 25% secreted by the parotid gland. The remaining 5% secreted by sublingual glands. What are the importance of the saliva? What's the function of saliva? Saliva help in lubrication and cleaning the mouth cavity, participates in taste sensation, also has antibacterial activity as saliva rich in immunoglobulin A, also helps in initial digestion by salivary amylase and lipase. In some non-human species, saliva has evaporative cooling function. Saliva also has buffering function, forming protective coat on teeth. Also participate in clotting process and wound healing. Now let's know the general structure of salivary gland. As any parenchymatous organ, salivary gland it is formed of stroma and parenchyma. Stroma consists of a capsule, which is the outer covering of the gland that sends trapeculae or septa, which divide the gland into lobules and reticular connective tissue. Reticular connective tissue formed min cells and fibers they form a network supporting the parenchyma. The parenchyma formed of the functioning cells. The functioning cells organized here as secretory units and duct system. Secretory units and duct system. This is the capsule, outer covering of the gland. From capsule, trapeculae extend into gland, dividing it into lobules. And stroma also consists of reticular connective tissue, which is formed of cells and fibers, form a network, support the parenchyma. The parenchyma formed of the functioning cells of the organ, organized in salivary gland as secretory units and duct system. This stroma of the main salivary glands, it is formed of the capsule, which is the outer covering of the gland. It is formed of dense connective tissue containing the blood vessels, the lymphatics, and nerves, which supply the gland. From the capsule, septa or trabeculae extend, divide the gland into lobules. Divide the gland into lobules. This is the septa, and this is the lobule. This septa. Also, it is formed of reticular connective tissue. Reticular connective tissue formed of cells and fibers, forming network. This network supports the parenchyma. Now let's know the structure of the parenchyma of the main salivary glands. The major or main salivary glands parenchyma formed of secretory portion or unit, which is formed of cells, groups of cells arranged in the form of acini or alveoli or tubules, surrounded or invested by myoepithelial cells. Also formed of duct system. This is the secretory units and duct system that will transport the secretion to the outside of the gland. And the secretory portion formed of these cells that will form the secretion. This secretion may be serous or mucus or mixed. So the secretory portion may be formed of serous acini, mucus acini, or mixed acini of both serous and mucus. The duct system or portions highly branched and range from small intercalated ducts to very large main ducts. Duct system that carry the secretion from the secretory portion or unit to the outside of the gland, to the mouth cavity. As we mentioned, that the secretory portion formed of a group of cells arranged in the form of acinus or alveolus. And these cells form the secretion of the gland. And the secretion may be serous or mucus. Now, let's know the structure of the serous acinus. The acinus, which is formed of a group of cells that form serous secretion. The serous acinus has a narrow lumen, and they are small in diameter. These cells pyramidal in shape. The cells are pyramidal in shape with rounded basal vesicular nucleus, basophilic striation or cytoplasm, apical acidophilic zymogen granules. As you see in the picture, pyramidal cells with basal, oval, rounded nuclei, apical acidophilic zymogen granules, and basal basophilia. The serous acini surrounded by few myoepithelial cells. We will discuss myoepithelial cells later. By electron microscope, serous cells have the character of protein secreting cells. They have abundant basal rough endoplasmic reticulum, prominent supranuclear Golgi complex for modification of the protein formed by the rough endoplasmic reticulum, numerous membrane-bounded spherical secretory granules containing the secretory proteins which are the enzymes as amylase, and they also contain numerous mitochondria. Now let's know the structure of this mucus acinus. Mucus acinus is formed of a group of glycoprotein secreting cells. Mucus acinus have a large diameter and wide lumen. These cells have flattened basal nuclei, pale basophilic foamy cytoplasm due to dissolved mucin granules. So it appears as foam. Mucus acini surrounded by numerous myoepithelial cells. Myoepithelial cells have contractile function, help drainage and squeeze the acinus to discharge the secretion out into the ducts. The content of the mucus in the saliva is very important for moistening and lubricating function. The mucus contains glycoproteins, which are formed by the well-developed supranuclear Golgi, which can be seen by an electron microscope. So, by electron microscope of glycoprotein secreting cells of the mucus acinus, we can see basal rough endoplasmic reticulum, supranuclear Golgi apparatus, numerous membrane-bounded mucin granules, numerous mitochondria. Let's summarize and compare between the two main types of the acini of the secretory portion, serous acini and mucus acini. Serous acini have a small diameter and narrow lumen, while mucus acini have larger diameter and wide lumen. Cells of serous acini have rounded basal and vesicular nuclei, while nuclei in mucus cells are basal and flattened. The cytoplasm of the serous cells show basal basophilia with apical acidophilic zymogen granules, while mucus cells show pale basophilic foamy cytoplasm due to dissolved mucin granules. The serous acini are surrounded by few myoepithelial cells, while mucus acini surrounded by numerous myoepithelial cells. This is due to the nature of the secretion. The mucus is thick and viscid, so it needs numerous myoepithelial cells cells having contractile function help squeezing the secretory unit and help discharge of the secretion out into the duct system. By electron microscope, serous acinous cells have protein secreting character with abundant basal rough endoplasmic reticulum, prominent supranuclear Golgi complex, numerous membrane-bounded spherical secretory granules, and numerous mitochondria. Mucus cells have character of glycoprotein secreting cells. They have basal rough endoplasmic reticulum, supranuclear Golgi with greater carbohydrate component, numerous membrane-bound mucin granules, and numerous mitochondria. As we mentioned before, the function of serous acini secrete watery saliva rich in amylase and lysozymes, while mucus acini secrete mucus secretion, thick and viscid, containing glycoprotein, which is important for moistening and lubricating function. Let's know the structure of mixed acinus. What is the structure of mixed acinus? As we see in this picture, mixed acinus formed of mucus acinus capped by aggregation of serous cells. They called it serous demilion. Serous acinus capped with aggregation of serous cells. Myoepithelial cells or basket cells, they called basket cells because they have numerous processes surrounding secretory cells like a basket. They are contractile cells having filaments, contractile filaments, actin, myosin. They lie between the plasma membrane oval secretory cells and the basal lamina, forming hemidesmosomes with it. Also, they are found around cells of initial part or proximal part of the duct system. They have a contractile function. When the process contracts, they press on acini, facilitating the release of secretory products into the ducts of the glands. This diagram shows the secretory units, which is formed of either mucus acini or serous acini, and the duct system starts with the intercalated duct, then striated, and shows also a myoepithelial cell surrounding the acinus. Myoepithelial cells with their processes form a basket surrounding the acinus, and also the initial or proximal part of the duct system. Let's now know the structure of the duct system. Duct system highly branched and range from small intercalated ducts to very large main ducts. It includes interlobular ducts, which are located inside the lobule, interlobular ducts located between the lobules and the main duct. Open to the surface in salivary glands. They open into the mouth cavity. The interlobular ducts include intercalated ducts Then the secretory duct, which is also called striated duct. The intercalated ducts lined by single layer of small cuboidal epithelial cells. They are surrounded by some myoepithelial cells. Several intercalated ducts join to form the striated ducts. The cells of the intercalated ducts may act as stem cells. They have the ability to divide and differentiate into secretory or ductal cells. While the structure of striated ducts or secretory ducts, as you see here in a light microscopic photo, cells the duct lined by a single layer of cuboidal to locolaminar epithelial cells. They have acidophilic granular cytoplasm with basal striation and central rounded nuclei. By electron microscope, cells show the character of ion transporting cells with basal plasma membrane enfolding and basal mitochondria. This is because these cells involved in modification of electrolyte composition of saliva. So the functions of striated ducts, the cells have the ability to modify electrolyte composition of secretion formed by the acinar cells. Forming secretory saliva. Also, striated ducts secrete lysozymes which attack bacteria and also involved in transport of immunoglobulin E secreted by the plasma cells which are present in the surrounding connective tissue and secrete into the lumen. Okay, let's know the structure of excretory ducts. As we mentioned that secretory system is highly branched and ranges from small intercalated ducts to very large main ducts. Also, the lining epithelium will change gradually from low to high epithelium with the gradually change in the diameter of the duct. We mentioned that the smallest ducts, which are the intercalated ducts, lined by small or low cuboidal epithelial cells. Then the striated ducts lined by cuboidal to locolaminar epithelial cells. So when we mention the interlobular ducts, they range from small to large. Start with colaminar epithelium, then pseudostratified colaminar, and the distal parts lined by stratified colaminar epithelium. Then the main ducts of major salivary glands, their proximal part lined with stratified colaminar epithelium, and near an opening, their orifice into a mouth cavity, they become non-keratinized stratified squamous epithelium. Okay, as we mentioned that major salivary glands are paired glands, including two parotid glands, two submandibular glands, and two sublingual glands, and they possess long ducts that open into the oral cavity. Now we are going to discuss parotid gland. As we see here, this is parotid gland open with long duct into the mouth cavity. Parotid gland located subcutaneously on either side of the face, below and anterior to the ear. They are purely serous compound tubuloalveolar glands. They have thick, well-developed capsule and thick interlobular septa containing many lymphocytes, plasma cells, and numerous fat cells. They show pure serous acini and prominent striated secretory ducts. Parotid glands produce about 25% of total salivary secretion. This is a light microscopic photo of parotid gland showing the major duct with a large lumen, serous acini with small diameter and narrow lumen, and intralobular ducts inside the lobule. This diagram also shows parotid gland with septa dividing the parenchyma into lobules, with serous acini, the secretory portion, and the duct system in between. In this photo, we see the serous acinus lined by serous cells, pyramidal in L shape, with apical acidophilia and basal basophilia, rounded nucleus. Notice the lining epithelium in the duct system, starting with intercalated ducts. The epithelium lining the intercalated ducts is low cuboidal, then become high cuboidal or low columnar in striated ducts. Then in interlobular ducts become columnar in the smallest, then become pseudostratified columnar, then stratified near the main ducts. The submandibular glands located on either side between the mandible and the muscle, forming the floor of the mouth. They are mixed compound tubuloalveolar glands. They have thick connective tissue capsule, but they have thin septa, showing less fat cells. The acini are mainly serous, forming about 90% of the acini. There are a few mucus and mixed acini. Submandibular salivary gland produce about 70% of the total saliva. This is a light microscopic photo of submandibular salivary glands. In this photo, we can see serous acini, small diameter and narrow lumen, acidophilic cells. While in this part, we can see mucus acini, larger in diameter with wider lumen. The cytoplasm of these cells, as we see, basophilic foamy, the nuclei flattened and basal. Some acini we can see may be mixed, as you see in this one, may be mixed acinus. Also, we can see the duct system, as you see here. This striated duct show basal striation, rounded central nucleus. Also here, striated duct, then the excretory ducts can see in septa between the lobules. Finally, the sublingual salivary gland. These glands are located deeply in mucous membrane of the floor of the mouth on either side of the vernilium. They are mixed compound tubulo-alveolar glands, contain numerous mucous acini forming 60% of total acini, with some serous acini. They are predominantly mucous gland with some mixed acini. They have less developed thin capsule and well-developed connective tissue septa. Few less developed intercalated and striated ducts are seen in submandibular glands. Submandibular glands produce about 5% of total salivary volume. Okay, finally, let's summarize and compare between the three types of the major salivary glands: parotid, submandibular, and the sublingual gland. Parotid gland, as we mentioned, they are located subcutaneously on either side of the face, below and anterior to the ear. The submandibular gland located between the mandible and the muscles of the floor of the mouth, while the sublingual gland, they are located in the floor of the mouth on either side of the vernilium of the tongue. When we mention the structure, the stroma of each gland. In a parotid gland, the capsule and septa are thick and fibrous with fat cells. The stroma in submandibular gland, the capsule is thick and the septa is thin with less fat content. In sublingual salivary gland, the capsule is thin and the septa is thick. Okay? In parotid gland, the septa and capsule thick. Submandibular gland, the capsule thick while septa thin. In sublingual gland, the capsule is thin while the septa is thick. As regards the parenchyma, parotid gland is a purely serous gland. Purely serous, contain serous acini only. The duct system show prominent intralobular ducts, and the main duct opens opposite the upper second molar teeth, called Stenson's duct. The submandibular gland predominantly serous, about 60%, with some mucus and mixed acini. The duct system show prominent interlobular ducts, and the main duct open to the floor of the mouth and is called Wharton's duct. The submandibular gland, the parenchyma of a sublingual gland, is a predominantly mucus with some serous demilions. The duct system show few intralobular ducts, and the main duct opens by several ducts into the floor of the mouth, ducts of Raven's. Okay. The saliva produced by parotid gland about 25%, 70% produced by submandibular gland, while only the remaining part 5% produced by sublingual gland. Okay, the first question say which salivary gland is located in front of the ear? Parotid gland, sublingual gland, submandibular gland, minor salivary gland. Yes, exactly. This parotid gland, a sublingual gland located deeply in mucous membrane of floor of the mouth on either side of the verulium, while submandibular salivary gland located on either side between the mandible and the muscle, forming the floor of the mouth. While minor salivary gland like labial or lingual in the lip and in the tongue. So the salivary gland, which is located in front of the ear, it is the parotid gland. Okay, second question. Which salivary gland is responsible for producing the majority of saliva in the mouth? Parotid gland, sublingual gland, submandibular gland, minor salivary glands. Okay, the major salivary glands produce the saliva, the majority of the saliva. What are the major salivary glands? Parotid, sublingual, submandibular. Okay, parotid gland produce about 25%, while sublingual gland produce about 5%, while submandibular salivary gland produce about 70% of the salivary volume. So, submandibular gland is the major salivary gland that responsible for production of the majority of the saliva. Okay, let's take another question. Which salivary gland has thin capsule and thick septa? Parotid gland, sublingual gland, submandibular gland, minor salivary gland. Okay, parotid gland has thick capsule and thick septa. Submandibular salivary gland has thick capsule and thin septa, while sublingual salivary gland has thin capsule and thick septa. So the answer is sublingual salivary gland. Okay, next question. Which salivary gland is purely serous gland? Parotid gland, sublingual gland, submandibular gland, minor salivary gland. Okay, purely serous gland, it is the parotid gland. The answer is number A, parotid gland, as sublingual gland predominantly mucous gland and submandibular gland predominantly serous but not purely serous gland, while minor salivary glands mostly they are mucus secreting glands. Okay, thank you so much for your listening.